Welcome back, ladies and gents, to our series of lectures on methods. This is part 6, neuroimaging. So we talked about a bunch of methods that don't involve neuroimaging, and now we're going to talk about a bunch of methods that do. So this is where we left off in the last lecture. Okay, these are all of our different sort of big groups of methods, approaches that we take to connecting cognition to brain. So let's look at some of these sub-approaches in neuroimaging. And when we talk about neuroimaging, there are two major categories, okay, that the methodologies are focused around. Right? The first is structural. Okay, so we want really good pictures of the brain, pictures of how things are built and how they're connected, right? That's the structural stuff. But we also really want to know about functional stuff. Functional stuff is like, how do these parts of the brain work together? Can we see the activation going on in this part of the brain? So these are what the neuroimaging techniques are really focused around capturing. So let's start at the beginning. It's a good place to start. Let's talk about computed tomography, or CT scanning. And this is essentially x-rays, right? And you sit in this cool little contraption here, and they take some x-rays, and the x-rays essentially so the scanner is going around your body. Okay, so it's taking x-rays in all different directions of your body, and it essentially builds a tomographic image, which is a slice. So you have a bunch of slices, and when you put all these slices together, when you stack them all up, you have a 3D representation of the brain, right? So individual slices are two-dimensional. When you have lots of slices, then you can have a three-dimensional representation of the brain. Now, CT scans have very high spatial resolution. They're great for seeing, bleeds in the brain, any kind of damage that might have happened from a traumatic brain injury. So really good spatial resolution. Structural or functional? What do you guys think? Is this going to be a structural technique or a functional technique? CT scan is all about structure. We want to know how things are built, and CT is a great way of getting at that. So now let's look at electroencephalograms, right? That's a big word. We'll just call it EEG. And EEGs are the ones that use these funny little caps, and they put all these electrodes on the surface of your scalp. That's what all those little doohickeys are electrodes. And these electrodes detect electrical current coming off of your scalp. You can have up to 256 nodes or electrodes on a cap. At least I think that's the max right now. So why electrical current? It's pretty good methodology for capturing what's going on in the brain, and that's because your brain actually runs on about 10 to 15 watts of power, right? So a night light bulb is about 7.5 watts. Okay, so you can actually measure the currents coming off of the brain. We will talk about how neurons work, and you will understand why we actually have electricity going on up there in a couple of lectures. Okay, so EEG is a direct measure of neural activity. Now, what do I mean by direct versus like indirect? So EEG measures brain responses that are a direct result of brain activity. Right? So you have brain activity that produces an electrical charge. You are directly measuring that brain activity. Now, indirect measures we will talk about in just a few slides, and you'll be able to see the difference. So in an ERP, you have an event, right? And you measure the response to that event with your little electrodes. So there are certain responses that research has discovered that correlate with kind of specific types of stimulation, right? So it's all about the timing in these kinds of responses. So when you're doing an ERP at about 170 milliseconds, there's a negative 170 spike, if you will. Think of it as something like this, okay? negative spikes are actually positive and positive spikes are actually negative. In ERP, it can be very confusing, but bear with me. So at about 170 milliseconds in the ERP, that is your brain processing phases. At about 300 milliseconds, this is where we make a lot of our decision making happen. Right? So that's where those processes are going on. We can do sleep studies with electroencephalograms. We can do seizure observations with EEG. So you can see here, this would be more like a comparison of what's going on in more conscious states versus less conscious states of awareness, right? Huge change in the kinds of brain waves, electrical activity that we see going on. And here, right, this is an observation of seizures. So up here, fairly normal brain activity coming from these particular spots, right and left. So left temporal, left frontal, left occipital, right occipital, and they do the same things with these measurements down here, except during a seizure. And these are the differences in results with the EEG. So some great techniques here at getting at what's going on in the brain. So what do you think? Is this going to be structural or functional? EEG is all about the function. So we want to know, while doing a particular task, does this part of the brain respond? Is this part of the brain involved in that function? Okay. Now, MEG is kind of just another take on EEG. So it's relying on the same source signal as EEG, right? So magnetoencephalography is not necessarily looking at the electricity itself. It's looking at the magnetic fields produced by those electrical currents. So it's still a direct measure of neural activity because it's looking at the actual neural currents, right, and their magnetic fields. It has the same kind of clinical applications and experimental uses as EEG, but here you can see that this is like a huge, crazy, like almost like one of those cheese head hats sort of things. Like it's like this huge thing, and right. So EEG is just the cap with the electrode. So this is sort of one of the downfalls of MEG. It's very expensive to do, but it's less cumbersome. Those EEG caps, if you've ever been in an experiment with EEG or you've ever put an EEG cap on someone, they are a pain to put on because you have to get them on there, you have to get them under the hair, you have to make sure everything's working, all the nodes are actually capturing some signal. It can be very cumbersome. This guy, not so much, but it's a trade-off. So what do we think? MEG, doing the same kind of thing as EEG is doing. So it's probably going to be functional. Definitely a functional type of approach. Moving on to positron emission tomography. Now remember we were talking about the tracer dyes before, and this is where we use radioactive tracers. That sounds really scary. I'll explain it in just a second. Combined with imaging to give us information about what's going on in the brain. So with PET, you inject a radioactive isotope. So we're going to use raclopride as an example. Okay, raclopride is a sort of a synthetic compound that mimics dopamine. Well, no, it's a synthetic compound, but what it does is it binds to dopamine receptors. So it doesn't necessarily mimic dopamine, but it binds to those receptors. When dopamine is around, okay, those receptors are full. So raclopride won't be able to bind to those receptors. So when you look at a particular area of the brain and you're following how that raclopride is traveling, right, you'll see a whole lot more clumping of raclopride in places where it can't be absorbed by the receptors. And you'll know that there's a lot of dopamine activity going on there. Hopefully that made sense. It's a little difficult to do, with audio only. But essentially what you're doing is you're tracking neural activity via the tracer. Now, we talked about direct measures of neural activity. This is an indirect measure of neural activity because, as I just told you, you are assuming the presence of dopamine because of what raclopride is doing in the neural synapses. Okay, so you are not actually measuring neural activity. You're making an assumption about what should be going on there based on what you're seeing. That's an indirect measure. Now, PET is cool, but it's also very expensive and it's also very invasive. It does, however, have excellent spatial resolution. It's just not very great temporally. So if you want to, for instance, track the feed-forward processes and the sort of the timeline during a particular cognitive task, you're not going to use something like PET because it's not going to have a very good temporal accuracy, if you will. So here's an example of what we can get using PET. So here we have someone passively viewing words, right? They're using their occipital cortex to just look at words. Here they are listening to words. You've got Wernicke's area is super duper happy right now because Wernicke's area is where we comprehend language. We have people speaking words. So now we've got some primary motor cortex activity here. We've got some anterior, inferior frontal lobe going on. That is Broca's area. That's where you produce language from. And then we also have generating verbs. So we have you're using your syntactic store, kind of. This is where language is in order to produce words from Broca's area, right? So this is all sort of in line with what we know about how the brain works, and PET shows us these things very nicely. So if it's showing us how things function in the brain while we're doing a particular task, it must be really good for functional, right? It must be a functional approach to neuroimaging. So let's get into the kind of the pop stars of neuroimaging. This is your MRI and your fMRI, your functional MRI. So MRI, we'll start with magnetic resonance imaging, is essentially you are in a giant magnet. Okay, you're in a giant magnet and it's producing radio frequency pulses. So I'm going to try and, in short, explain to you kind of how an MRI works. So it uses differences in tissue magnetization. Now, how does it do that? Okay, follow me here. We are made of a lot of water, right? We contain a lot of water. Therefore, we contain a lot of protons, right? Lots of water, lots of protons. The magnet, essentially, with its resting magnetic field, encourages our protons to align and spin with the field, right? So they all sort of like, it's like when your hairs stand up on end when you get really freaked out. They all kind of these protons just stand up and align themselves with the magnetic field. Now, you use a radio frequency pulse, boom, pulse. These protons, they all lay back down. Radio frequency pulse they just lay back down. And now, based on where these protons are, based on what kind of tissue they're in, they will relax back to their standing state and realign with the magnetic field at different rates. Okay, so in some tissue, the protons are going to stand right back up a lot faster than in other tissue. Now, it's by measuring these differences in proton activity between these tissues, in other words, the magnetization properties of the tissue, by comparing and contrasting those things, that's how we get these really cool images in fMRI. Now, hopefully that made even just a small amount of sense, but if you have questions, ask me and I will explain it. Okay, so really awesome spatial resolution with fMRI, like really, really, really great. So when we combine fMRI with some different techniques, right? So if you do different kind of pulsing techniques in the magnet, you can get measures of different kinds of activity. So with fMRI, the F stands for functional, and essentially what you're doing is combining what's happening in a regular MRI and what we call the bold signal. Okay, and this is the blood oxygenated level dependent signal. Whoa. So when different tissues in the brain return to equilibrium, there are, okay, let me start over. So when we talk about bold, active neural tissue uses nutrients in the blood. Okay, so when it's using nutrients in the blood, it's sucking up all the oxygen from the blood. So deoxygenated blood is a signal that there's a lot of neural activity going on there. Essentially, this is what fMRI is doing. Okay, it's using the spatial resolution awesomeness of the MRI approach and adding in this extra measure of finding where or comparing blood oxygenation levels across brain tissues. Okay, so that is the additional kind of kick that fMRI brings to the situation. So, of course, great spatial, just like MRI. Pretty good temporal, not like super awesome, but it's pretty good. So it can take an image every 2 seconds, like one full image of the brain. So let's say you want 36 slices of the brain, right? You want to get 36 separate pictures, and when you stack them on top of each other, you'll have a 3D representation of the brain. Okay, it can take those images, those 36 images in 2 seconds, right, and combine them all for a 3D representation of your brain, in short. Okay, so fMRI can tell us really great things about stuff like functional specialization, right? We like functional localization. We've been talking about it lately. Are certain brain areas specifically designated for certain cognitive tasks? So, for example, the fusiform face area is pretty popular in the fMRI literature and the history of fMRI. It's critically involved in processing faces. So I'll show you here. In this first image here, we have faces being shown, upright, intact faces being shown, and we have objects being shown, right? So we're looking at signal change in this fusiform face area. When you have a face, lots of signal. Object, not so much. Face, lots of signal. Object, not so much, right? Now, when you look at intact versus scrambled faces, here's your intact faces and scrambled faces. When this area saw an intact face, it was really happy. When it saw a scrambled face, it wasn't happy. When it saw an intact face, it was really happy. A scrambled face, not happy, right? So this area has this really general preference for faces, and particularly intact, upright faces. So these are the kinds of things that functional imaging can really tell us. So I, just gave it away. Functional imaging, fMRI is definitely functional, and MRI itself is actually structural. Okay, you're not deducting function from. I'm sorry, you're not deducing function from an MRI image. Excuse me. Okay. Excuse me. Last one. Diffusion tensor imaging. This is the technique that gets us these really cool pictures of the brain. It's an MRI technique, okay, but like functional MRI, when we add in that oxygenated blood measure, what we're doing here is using directionality and magnitude of the diffusion of water along myelinated fatty neurons. Okay, that was a whole lot, right? Okay. When we talk about neurons, you're going to learn about this stuff called myelin. Myelin is essentially a fatty covering that runs along the length of the axon of the neuron. Okay, it acts like insulation. Now, because we're full of water, right? Water, it tends to diffuse along the fatty axons, and what you can do is sort of follow that water. So if you follow the water, it will actually tell you, like, here's where this particular axon is going, and you can make all of these cool little projection traces. Okay, you'll see a little bit more about what we're talking about when I show you like what neurons look like and how they're made, etc. So you essentially, we use fractional anisotropy. So you take the shape of the diffusion tensor at each voxel. So a voxel is a tiny little square, a tiny little cube of the brain that we want to look at one particular time. So I want to look at what this little cube area of neurons is doing at this particular time. So you take that cube and you look at the direction that the water is going in that particular cube. Then you look at the cubes right next to it and you look at the direction the water is going there. So then you can sort of deduce by adding this information together how the water is traveling, right? So again, this is an indirect measure of brain structure and brain activity. So it has a really great clinical application in that you can really look at connectivity after a stroke or an accident, and connectivity is very important. Remember, I said you can cover your eyes and assume that your eyes are completely responsible for vision, but really that's not the case, because your eyes are connected to a huge system in there, this huge visual system that takes care of a whole lot of different things, way beyond what your eyes are capable of doing. So, connectivity in the brain, very important. So that was our DTI, mainly going to be a structural technique. Okay, you're looking for, which way neural tracts are going. So there's not a whole lot of function being sought out with diffusion tensor imaging. So as a last sort of reference for you, this is kind of so if we're looking at temporal goodness, okay, like how good are all of these methods on a temporal scale, and how good are all of these methods on a spatial scale, right? So let's look here. Light microscopy, right? It's on the level of like minutes as far as temporal resolution, but it has a really fine spatial resolution, right? You're getting down to neurons, dendrites, and even maybe possibly at the area of the synapse with this kind of imaging versus lesion studies. Temporally, no. Okay, and spatially, you're really only going to get at the whole brain level of things. Okay, so micro lesions here, which is sort of a newer method, which is definitely a newer methodology, those can get a little bit finer on the spatial scale. So that's just, you can look at all of the different methods here and compare and contrast. This is just kind of like an FYI chart in case you're interested. Okay, so that is it for methods. I will see you in the next series of lectures when we talk about brains. So that'll be fun, and I will see you guys there.